All right, so here is my attempt to help review a little bit part of the AP test. So what we're going to do today is we're going to talk about DNA replication and how it applies to the universal code. Um, of course, in honor of that, I got to wear my Rosalind Franklin shirt to represent our girl. And of course, we remember the fact that she was not the only person instrumental in discovering the structure of DNA. That there were a lot of moving parts in that, and she was one of them, as was Maurice Wilkins, and Watson and Crick put it all together. So while I definitely honor her contribution, I have to acknowledge that she was not the only factor, just as a little bit of a reminder. So let's start going over some of these things. See if I can't move this just a little bit closer. All right, hopefully that helps. So when we're talking about replication, there's a few things that I definitely want you to remember, okay? I think you need to remember the enzymes. It's always important to remember what an enzyme is and what it does. That's super important, of course. It's also important to remember what it's made of, which would be tertiary and quaternary structured proteins, and what could disable it either temporarily like an inhibitor or permanently like heat, pH, or salt. But the enzymes that you need to know for sure for AP would be these. So topoisomerase is what actually like untwists or uncurls the double helix. Helicase is everybody's favorite memes. You know that this is what actually unzips the genes. Specifically, it's going to unzip or break some of those hydrogen bonds, and it's going to hold the place as it goes. Primase, it's kind of a generic name. Primase is going to attach a primer to the parent strand, the original strand, and then that's going to add a little RNA primer to that strand. So then the, in this case, DNA polymerase can attach to that to go ahead and continue creating. We've got DNA polymerase here, and it's going to have two major jobs. Now remember, there are several different DNA polymerases, so I'm using a generic DNA polymerase like category of enzymes here. So the first one is that it's making DNA polymers by adding nucleotides, and of course that is according to Chargaff's rule. The second job, which is also very important, is that it is spell checking the DNA. And so that's going to ensure not that mutations don't occur, but that every T is bonded to an A, every C is bonded to a G. So when it works correctly, and it does most of the time, it's pretty effective, it's going to make sure that we're not introducing new mutations through mismatch. Okay? And then the last one we have is ligase. Remember, that is something that's holding tight like a ligature. And so we say that it glues the pieces of, in this case, the DNA together. Okay? As a little reminder, and we'll talk about transcription a little bit later, DNA polymerase is used in replication, but RNA polymerase is used in transcription. So the enzymes are pretty similar for both processes. The difference is what type of polymer you're making. Okay? All right, so let's talk a little bit about structures, because we know that structure and function go together. So just as a little review, see if I can't get this in view a little bit better. There we go. Okay, so we have the nucleotide, which of course has a phosphate group. It has a 5-carbon sugar, which in this case is going to be deoxyribose, and then it has our nitrogenous base, which is A, T, C, or G. Excuse me. So when we're looking at that, those are the 4 nucleotides that we would see in DNA. Of course, in RNA, instead of the T, you have a U. We remember that the phosphate group is negatively charged, and that is what makes DNA also negatively charged. This is significant when we're talking about things like how we use gel electrophoresis to show DNA movement when we add electricity to it. So that's an important facet of DNA that allows us to do a lot of biotech type work. We remember Chargaff's rule, that A is equal to T and C is equal to G, and of course that was in amounts or percentages in his time, and Watson and Crick identified it as A was bonded to T, C is bonded to G with hydrogen bonds, and just as a quick FYI, there are 2 between A and T and 3 between C and G, and that's why I always make the equal signs like that, just so you and I all remember. When we're looking at the polymer of DNA, so I've got a very short polymer. Remember the 5' is the phosphate side, and it's always going to be bonded to, hydrogen bonded to the 3' on the opposite strand. So we've got 5' on the top and 3' on the bottom of the little house, okay? And it's flipped on the other side. So we use the term anti-parallel, which is the term that Watson and Crick coined for this. And then just an FYI, I don't think it's super duper important for an AP exam, but just if you see it, don't panic. Glycosidic is the bond between the sugar and the phosphate, so like right here, the bond that keeps the nucleotides together, right there. I just know that when you look at glyco, you think glucose, and in this case, it's just referring to the generic sugar. Okay, so some basic things to remember about the structure, because that determines a lot of things about the function. So when we're talking about Watson and Crick's contribution, one of their major contributions was understanding that the structure of DNA was going to determine a lot about replication itself, which is completely valid. So when we're thinking about it, one of the other main things that you need to remember is the concept of the leading and the lagging strands of DNA. So we've got a strand here, and I'm not showing all of the bases, but understand that the bases are there. I'm just trying to simplify this sketch for you. We have a helicase, so topoisomerase has come in, unwound a bit. Helicase is opening that little bit. DNA continues further down. So here is the replication fork is heading in this direction. We have DNA polymerase attaching to both sides, and it's going to attach so that it's going to make a new strand in the 5' orientation. So this is 3', so this is going to be 5'. This one here, that's 3', so this is 5'. DNA polymerase is going to attach there. From there, it's going to move in this direction. This one's going to move in this direction. Okay? So things to remember, replication only occurs from 5' to 3' on the new strand. And the fact that it's on the new strand is super duper important. That took me a little while to figure out because no one actually flat out said it. It was just assumed that we figured it out, and well, it took me a little while to get it. So I want to make sure y'all got it. The leading strand, this one, is the one going in the same directions as the helicase. So it is moving with the helicase, so it's going to make a long strand. The lagging strand begins at the helicase and goes in the opposite direction. And that's going to cause chunks, because as the helicase goes up, it's going to make another chunk, and then it's going to move further, and it's going to make another chunk. And of course, we call these chunk Okazaki fragments in honor of the husband and wife team that discovered them. And these are going to be bonded later with a ligase to sum everything up. Okay, so some things that are super duper important to remember in general about this whole process and what it means. Again, let me see if I can't get that a little bit better. Okay, so we remember that it is the universal code. So DNA is common in all organisms and most viruses. Of course, there are viruses like HIV and COVID-19 that use RNA instead of DNA, but a good chunk of those viruses are DNA bound. And when we look at it, I'm using quotation marks on purpose, y'all. Always the structure is the same. So it's always a double helix. It's always 3' to 5'. Now I say always with the quotes because there have been some articles written in the not too distant past suggesting that they have found or have created 3-stranded DNA. However, that is in very, very specific circumstances. That is not common. So you are allowed to say always. I just want you to be aware that there is some literature to the contrary, so that when you go to college and you take your advanced biology courses, you don't come and tell me later that I'm a big fat liar, because that's certainly not my goal. Then we have this interchangeable concept between organisms and species. So because it's common, just like, I don't know, like Henry Ford assembly line-ish concept, if you have some common pieces, then you can interchange things between. So this part will work on the Model A and the Model T both concept. And we can do that with organisms. So it happens in nature through a process called transformation. Okay, and so that was discovered way back when by Griffith and his experiment. And we can do it intentionally in a lab. The process is still called transformation, but the organisms that are then created would be called transgenic, or in popular society, they would be called GMO. So it does happen in nature. It can also happen at the hands of humans. But the fact that we can interchange this is based on the fact that the DNA is common. The other thing to remember are some of the differences between pro and eukaryotic cells. And so this is a thing that comes back, a theme that comes back over and over and over through AP, is how they differ from each other. So when we're talking about the DNA and replication and things of this nature, let's talk very briefly about some of those. So of course, the first thing to remember is the location of the DNA in here. The prokaryotic cell is going to have its DNA in the cytoplasm. It does have a nucleoid region, which is like a general area where we can find the DNA, but it is not a true compartment. And then we have eukaryote, of course, which is going to be in the nucleus when we're talking about nuclear DNA replication, so the primary stuff, okay? And of course, in this case, our little analogy would be that eukaryotic is like having all of your notes and papers in your backpack that is then sorted into like binders and pencil bags and notebooks and things like that, so that you can go in and very easily grab what you need to do your work. Prokaryotic is more like when you just kind of grab everything that you have, your schoolwork and your personal stuff, and you just throw it randomly around your room, and you hope you find what you need before class. You kind of sort of normally throw it near your desk-ish, but it could possibly be somewhere else. So that would be the difference between the prokaryotic and eukaryotic. It's a matter of organization. Other things that differ: eukaryotic has multiple replication forks that happen simultaneously in each of the chromosomes, and it increases the speed of replication. So rather than doing top to bottom, each one individually, by doing multiple replication forks and then adding the pieces together through ligase, we're going to get through this much, much faster. Prokaryotic cells only have one piece of DNA. It's not quite a chromosome, but you could call it that and it would be all right. It's just not the most precise term, I guess. So it's one piece of DNA. It's normally much, much shorter than any eukaryotic like chromosomes, but there you go. And then they typically have a plasmid, which is that circular piece of bonus, I like to call bonus DNA. And that's frequently going to have things like resistance to various antibiotics, or the ability to, I don't know, glow in the dark and stuff like that. So that's one of those special little tools that we use because the plasmid doesn't affect the basic construction of the prokaryotic cell. It affects like all of the extra cool stuff that the prokaryotic cell does and has. Okay, so I'm hoping that cleared up a few questions. This would be the main concepts that you would need to remember to get ready for the AP exam. Of course, if you have additional questions, you can contact me by email or text, and I'm happy to answer questions that you have. Remember that when we are talking about the AP exam, we're not looking necessarily for all of the, highest vocabulary. We're looking for a deeper understanding. So if there's a word that you don't know or you can't remember the term, don't stress out about that specific term. Try to explain what you mean. Explain the process. So process is more important than vocabulary when we're looking at AP exams. And in most cases, not all because, I don't write all the rubrics, but in most cases that I have seen, being able to describe it, describe the process or describe the item you're talking about and what it does is frequently interchangeable than with using just the term itself. In fact, sometimes it's preferable so that you're explaining what you mean and not just regurgitating a vocabulary word. Okay, so I'm hoping this is helpful. I know that if Rosalind Franklin was still here, she would be cheering you on as much as I am as you're getting ready for this. I know you can do it. Let me know if you have questions. Mwah. Best of luck, y'all.